Hello everyone, and welcome to another tutorial for BioSE1A. This tutorial is about what is involved in photosynthesis. A new pesticide is being tested. The side effect of it is a decrease in reduction of NADP+ to NADPH in the chloroplast. Given this observation, what would you expect to happen when you measure production of 3-PG and RUBP in plants that are treated with this pesticide? Before we answer this, let's take a quick look at photosynthesis. Photosynthesis is an anabolic process. The energy of sunlight is captured and used to convert carbon dioxide into more complex compounds like sugar. So two things. One, capturing the energy of sunlight, and two, converting carbon dioxide and water into sugar. There are actually two reactions. The light reactions convert light energy into chemical energy in the form of ATP and the reduced electron carrier NADPH. This occurs in the thylakoid. The second reaction is the light-independent reaction, or the Calvin cycle, which does not use light directly, but instead uses ATP, NADPH, and carbon dioxide to produce carbohydrate. This happens in the stroma. For the light reaction, thylakoids contain a pair of photosystems called photosystem 1 and photosystem 2. In photosystem 2, chlorophyll molecules in the reaction center absorb the light energy, which excites electrons to a higher state. The excited electrons will be passed to an electron transport chain. The electrons lost by the photosystem 2 are replaced by photolysis, which involves the oxidation of a water molecule, producing free electrons and oxygen gas. As electrons pass through the electron transport chain, the energy from the electrons is used to pump hydrogen ions from the stroma to the thylakoid space, creating a concentration gradient. The movement of protons back to the stroma powers ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving the electron transport chain are shuttled to photosystem 1. The low-energy electrons are re-energized by another photons. The excited electrons are passed through an electron transport chain, where they are used to reduce the electron carrier NADP+ to NADPH. So for every excited electron, one ATP is produced from photosystem 2 and one NADPH is produced from photosystem 1. Those ATPs and NADPHs are used in the stroma to fuel the Calvin reaction. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce carbohydrates. The cycle consists of three steps: carbon fixation, reduction in sugar production, and regeneration of CO2 acceptor molecule ribulose 1,5-bisphosphate, or RUBP. In the carbon fixation, CO2 is attached to RUBP, resulting in a 6-carbon molecule that splits into two 3-carbon molecules called 3-phosphoglycerate, or 3-PG. The reduction step uses electrons from NADPH and some of the ATPs to reduce 3-PG to glyceraldehyde 3-phosphate, or G3P. In the final step, RUBP is regenerated. For every 3 turns of the cycle, 5 molecules of G3P are used to reform 3 molecules of RUBP. The remaining G3P is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of G3P to make 1 molecule of glucose. So Calvin cycle has to run 6 times to produce 1 molecule of sugar. Now back to the question. The pesticide that is being tested decreases the reduction of NADP+ to NADPH. What would you expect to happen when you measure production of 3-PG and RUBP in plants that are treated with this pesticide? So for the system 2, it's still producing ATP, but for the system 1, will not be producing NADPH. This means ATP is still supplied to Calvin cycle, however, there is no NADPH being supplied. This means the 3PG cannot be further processed into G3P. The Calvin cycle is stuck halfway through the reduction step, which means the production of sugar and the regeneration of RUBP are halted. So you would expect to observe an increase in the 3PG levels and a decrease in RUBP production. That concludes this tutorial. Thank you for listening and good luck.